Hello everyone, let's get started. Today we're going to be talking about measuring microbial growth. There are times in a microbiology lab when we're presented with samples of material that need to be analyzed in order to determine the number of microbial cells that are present per volume of that sample. For example, municipal drinking water has to be monitored periodically. Recreational bodies of water have to be monitored periodically. Samples of foods that are prepared at commercial facilities have to be monitored. Those samples have to be taken into the laboratory, and we need to determine our best estimate of how many cells are present in that sample in order to ensure that that sample meets regulatory laws about what's allowed and what's not allowed. So in this lecture, we're going to be talking about two different areas of this topic. We're going to talk about how microbial growth gets measured in a sample. We're going to talk about the different methods that can be used, and we're going to talk more specifically about using plate counts. And second, we'll talk about the environmental conditions that influence growth. We'll talk about the specifics of what microbes can and cannot tolerate in terms of the conditions around them for growth. And finally, we'll talk a little bit about food production and safety. When we want to count the number of cells in a particular sample in the microbiology lab, we can do it one of two general ways. We can do what's called a direct count, and we can do an indirect count. A direct count, like the name suggests, is where we literally take a portion of a sample, put it onto some kind of a microscope slide, and count the cells. Similarly, we can take a portion of a sample and put it into a type of machine called a cell counter and let the cell counter count the number of cells. That is a direct measurement because we are literally counting the number of cells in a sample. Indirect measurements are different. In an indirect measurement, what you're doing is determining something other than the exact number of cells. You're determining the amounts of some other cellular component and then using math to extrapolate back to a cell count. This can be done using what's known as viable plate counting and also by measuring the cloudiness in a sample or the turbidity. Let's talk about each one of these separately. Regarding microscopy, we're going to talk a little bit about what are called counting chambers. We'll talk about how light microscopes are used to do this kind of counting. And we'll talk about how fluorescent microscopes are used and what advantages and disadvantages these two types of microscopes offer. When it comes to cell counting and cell counters, we're going to talk about what's known as flow cytometry and compare it to what's a machine called a Coulter counter. Let's begin with the microscopy. If you put a sample that has microbial cells in it on a regular microscope slide and put it underneath the microscope, you can certainly count cells that way, but it's a little bit difficult because it's hard to know which cells you've counted and which cells you haven't counted yet. In other words, it would be very easy for you to make a miscount, to count some cells twice and to miss some cells altogether. For this reason, we use very specialized microscope slides called counting chambers to do this kind of work. Now, there is a type of counting chamber called a hemocytometer, which you may have heard of before. This is a specialized type of microscope slide that's used to count red blood cells and white blood cells in a blood sample. A hemocytometer is essentially a thick piece of glass that has had a series of grid lines etched into it using a laser. So when you place the sample onto the microscope slide and put a cover slip over it, and then you look through the microscope, you're going to see the cells dispersed over this grid. The grid lines and the grid areas help us do a more accurate count of the cells. Now, what you're seeing here, you can see one great big square that basically fills up the whole image. And then within that square, there are smaller squares. Here's 1, 2, 3, 4, 5, 6, and so on. And then within those squares, there are even smaller squares. You could even go down into a square that's very, very small, like that one or that one. It all depends on the size of the cells you're counting and your needs. Rather than counting all of the blood cells, for example, that you would see in this grid, what we do is we count a certain number of squares, and we use a mathematical formula then to extrapolate back to the whole sample. You might, for example, count all the cells in this square that I'm outlining here, and this one in the center, and these two down here. That's a common practice when you're counting blood cells. You measure these 5 areas by counting the cells, and then you use a mathematical formula to take that number and extrapolate it back to the whole sample. Hemocytometers are very common. You find them in a lot of different types of laboratories when we need to do these manual counts of blood cells. Now, we can also use these types of counting chambers to count microbial cells. We use a different type of counting chamber, though. It's called a Petrov-Hauser or pH counting chamber. The idea is the same, though. It's simply a special glass microscope slide that has had a grid etched into it. We're going to place our sample onto the microscope slide with a cover slip. And when we look through the microscope, we're going to see the cells dispersed throughout this grid. We're going to count a certain number of squares, and we're going to use math to extrapolate back to the whole sample. This sounds like a nice idea. It's actually quite manageable. It's not a difficult technique to learn how to do, but there are some drawbacks to it. Number one is it's a manual technique. Someone has to sit down and do this manually. So it's only really useful if you have a small number of samples to count. If you have a large number of samples to do, this would not be an efficient way to do it. Number two, while it's relatively easy to see, say, blood cells on a counting chamber, when we're talking about microbial cells, it's a whole different situation, and that's because microbial cells are so small. If you recall from our gram staining lab, in order to see bacterial cells in any kind of detail, you have to go to the 100x lens. That, remember, is a 1000x total magnification. It's difficult to use these counting chambers at that high of a magnification. They were really designed to be used at lower magnification lenses. When we decide to do a direct cell count using a pH counting chamber, we can do the actual counting using either a light microscope or a fluorescent microscope. Each one of these types of microscopy has certain benefits to it and certain drawbacks to it. When it comes to using a fluorescent microscope, remember, we're going to want to first stain that sample using particular fluorescent dyes. These dyes bind to particular materials within the microbial cells, and then when we take the slide to a fluorescent microscope, the microscope has the ability to shine light of a particular wavelength through the sample, and that light will cause the fluorescent dyes to fluoresce, to give off a very bright color. Fluorescent microscopes are great in the sense that they allow you to see things very, very clearly because of the bright fluorescent colors that these dyes give. But remember, fluorescent microscopes are very expensive, and so they're not found as commonly in laboratories as light microscopes. The same principles apply when you're doing cell counting, direct cell counting with a fluorescent microscope as with a light microscope, in that you're going to place your sample, your stained or dyed sample, onto the pH chamber, onto this specialized microscope slide. You're going to look through the microscope, and you're going to use those grid lines in order to count a subset of the cells. Now, there are a couple of real benefits to using a fluorescent microscope compared to a light microscope. Number one, by using fluorescent dyes that specifically bind to particular cellular components, it will help you distinguish the cells you're trying to count from other debris that might be in the sample. Remember, when we talk about cell counting, we're often talking about using real-world samples. Samples of municipal drinking water, samples that came from a recreational body of water, samples taken from a food production facility. So we don't expect to see only cells in these samples, but also a certain amount of debris. The fluorescent dyes will not tend to bind to the debris. They'll tend to bind to the cells. So it's very helpful for us to be able to distinguish the cells in one of these real-world environmental samples. On the lower right-hand side of this slide, you're looking at a specimen that was prepared for use with a fluorescent microscope. There are cells present, these bright green structures. These are eukaryotic cells. I know that because you can see a nucleus, a light blue nucleus inside each one of these cells. And then around those cells, there are these much, much smaller, sort of orangey red colored structures. These are microbial cells. Remember, bacterial cells, archaeal cells, they're going to be much smaller than eukaryotic cells. And you can see that because of the different colors used, because of the different dyes used, whoever's doing this counting process is going to be much better able to see which cells are microbes and which cells are eukaryotic cells or other pieces of debris. Microscopy and direct cell counts are a tool that we use all the time in laboratories. They're fast. You don't need any very particularly specialized equipment. There's no need to take your environmental sample and subculture it. There's no need to wait, in other words, for cells to grow in an incubator. And it counts microbes that, in some cases, we wouldn't even know how to grow in culture. There are some microbes that are out in the real world that would be very hard to culture in the laboratory because we just don't know enough about them yet. So by eliminating the need to create a culture and by taking the sample directly to a microscope, we have an advantage. One of the biggest disadvantages of using a microscope and direct cell countings is this. A lot of the time when we are doing real-world sample cell counting, we're looking at samples that are going to have a low density of cell number in them. For example, in municipal drinking water, it's important that we look for cells and that we count cells, because we insist on a certain level of cleanliness in our drinking water. So while we do allow for an occasional cell in drinking water, we want to make sure that the number doesn't go above that limit. Now that limit is very low. And if you think about it, let's say the limit were 3 cells per ml of water. It's going to be hard to see those 3 cells in such a relatively large volume of water. So whenever you're examining a sample that you know is going to have a low density of cells in it, using a microscope is tough. You have to do something to try to concentrate your sample before you take it to the microscope, and that adds an additional level of complexity. Now, let's switch gears and look at the machinery that we have available for direct cell counting. The first type of cell counter is called a flow cytometer. Over here on the right side of the slide, there's a diagram that shows you just very basically how flow cytometers work. These machines have a place where the sample can be added into the machine, and then it has a network of tubes that are going to carry the sample and the cells past a light source. It's a particular type of laser that emits light of a certain wavelength. And this light is going to interact with the sample as it passes by, and it will interact with the cells if they're present in the sample. So sometimes this light, depending on the cell, will bounce off of the cell, and that will be an indication that the cell is present. Sometimes it will be absorbed by the cell. That will be an indication that the cell is present. It depends on the type of cells that are being counted. So this machine has to be set. It has to be calibrated to count a particular type of cell. And of course, cells vary by size and by shape. So flow cytometers work best on a limited number of cell types. There are some cells that simply cannot be detected by a flow cytometer. Many of our microbial cells fall into that category. The flow cytometer cannot count them accurately because they're just too small. Some of the cells pass by the light beam and are missed. They don't get counted. The other problem with flow cytometry is just that flow cytometers are expensive, so they're not going to be found in every single laboratory. It's an expensive piece of machinery. But you do find flow cytometers in certain laboratories that are trying to do certain types of cell counting. A good example is laboratories that evaluate semen samples, sperm samples, for reproductive purposes. In veterinary medicine, we do a lot of flow cytometry when we are trying to count sperm numbers in breeding animal samples. Once a year, we take a sample of semen from these male animals that are going to be used for breeding, and we do a full analysis of the sample to make sure that the sperm count is high enough to get the female animals pregnant. Flow cytometry works great when you're doing sperm counts. It doesn't work as well, though, for microbial cell counts in many instances. The second piece of machinery that we use for direct cell counts is called a Coulter counter. Coulter counters are especially common in large diagnostic laboratories, including hospital laboratories. Coulter counters can be used for counting all the different types of blood cells, for example, that you would find in a human being. Now again, you have to program the machine for whether or not it needs to count the red blood cells or each of the different types of white blood cells. You have to calibrate the machine to know which cell type it needs to count. Coulter counters are really accurate, they're very efficient, and they don't require a human being to sit there and manually count the cells. So they're very good in a laboratory that does a lot of samples, like a hospital laboratory. The biggest problem with Coulter counters is their cost. These are very, very expensive pieces of machinery. They are much more expensive than flow cytometers. And so you're not going to find a Coulter counter in, say, a small doctor's office or a small teaching or research laboratory. These are going to be in large diagnostic laboratories.